Hi again. I decided to do a second video of the evening. Um, this is a very important topic and a new development in RVCL research. And so I'm going to do my best to convey the information clearly. I'm sure there are going to be questions that come up, and I want everyone to feel free to reach out to me directly with questions, and I will get back to you. Uh, So the topic relates to breast cancer risk in women with RVCL or RVCL-S. So for anyone who doesn't know what RVCL or RVCL-S is, because we may have viewers who are not familiar with this is a genetic disease that is inherited dominantly. 50% of family members are affected. And this disease affects the brain, the eye, the liver, and the kidneys in 100% of individuals who inherit the disease-causing mutation. What we didn't know until recently is that this mutation also creates risk of breast cancer in women, heightened risk. So I'm going to explain what that means, how this was discovered, and what are the implications of this going forward. So why would patients with RVCL be at greater risk of breast cancer? Well, to begin to understand this, it's very important to understand the process of DNA damage. In every cell in all of our bodies, whether we have RVCL or not, there are lots of chromosome breaks that happen throughout the day. In just one cell, there can be 10,000 DNA breaks where chromosomes fragment over the course of a single day. And there are proteins that are almost like little molecular policemen, which can bring the DNA back together. Now, why do these DNA breaks happen? Well, there are environmental insults that trigger injuries to our chromosomes. It can happen from ultraviolet light. There's certain types of DNA damage that happen from ultraviolet light, chemicals, radiation, even something as simple as a CT scan can cause DNA breaks. Lots of CT scans can increase the risk of cancer. Replication errors in DNA. There could be metabolic issues that contribute to DNA damage. And so to understand how this relates to RVCL, first, it's important to know that we recently discovered that RVCL is a DNA damage syndrome. What that means is that chromosomal injuries are at the heart of what is driving this disease when we think in terms of molecular mechanisms, the things happening in a cell that eventually contribute to organ damage. There are issues in RVCL related to DNA repair and DNA breaks. Similarly, issues related to DNA repair create risk of certain types of cancer, including breast cancer. And so understanding that RVCL or RVCL-S is a DNA damage syndrome is at the heart, at the core of understanding the relationship between the organ injuries that occur in RVCL and the heightened risk of breast cancer, which we recently discovered in women with RVCL. So how do cells normally handle DNA damage? Well, there are a few different ways cells can handle DNA damage. There's a type of repair where two strands of DNA that have broken apart can be glued back together. This type of DNA repair is the kind of thing that happens in a radiation injury. You can almost imagine it like if you've broken a pencil in half and you taped it together so that it's usable again. It's not a perfect repair, but it's a repair that's sufficient to keep the cell alive and functioning. Now, this is the type of DNA damage that can occur in radiation injury. And remarkably, when one examines the brain of a patient with RVCL under a microscope, it looks indistinguishable from radiation injury. This is important to understand. Now, there's another type of DNA repair where the cell or the DNA strand borrows from another chromosome. So remember, we have pairs of chromosomes, one inherited from mom, another inherited from dad. Now you can imagine if you have a break on one chromosome, say the one you inherited from mom, the cell will borrow from the very same chromosome inherited from dad and make a perfect copy. So this is better than taping the pencil together. This is copying the paired chromosomes so that you bring a perfect DNA repair at that site of DNA break. And there are breast cancer-related genes involved in that process called BRCA1 or BRCA1, BRCA2. Now, interestingly, this type of DNA breaking and repair happens almost intentionally in a developing sperm or egg. It allows chromosomes of grandparents to shuffle in the developing sperm and egg so that a grandchild does not resemble exactly one of the two grandparents in terms of the chromosomes. It's a mixture of the grandparents' chromosomes. So that's where DNA breaks happen intentionally, one could say. And DNA repair is involved in shuffling of the chromosomes or the shuffling of the genetic deck. So there's different kinds of DNA repair, and breast cancer genes are related to this. Now, there is an entire theory of aging called the DNA damage theory of aging. So if I told you that these two mice are siblings and they're actually identical twins except for one gene that's causing DNA damage, that might be surprising because you can see that one of them looks much older than the other. They look very different from each other, even though they're identical twins other than one gene. And that gene in this case is one that is injuring the chromosomes. So this theory of DNA damage in aging is proven. We know that when chromosomes are injured, that cells begin to age more rapidly. And what we've discovered happens in RVCL is that certain cell types sustain more DNA damage, and certain cell types therefore begin to age more rapidly. This is a known consequence of chromosome injuries. Now, look at these cells. This is an experiment called a comet experiment, like Halley's Comet. And you can see in these experiments the DNA damage that is occurring in RVCL. On the left is normal TREX1. These are normal cells with normal TREX1. And on the right are cells that have the most common mutant or mutation that causes RVCL. And what happens here is the cells are in a gel and electricity is moved through these cells in a gel in a dish in the lab. And if the DNA is broken, you get a comet that forms. You can see how these almost look like a comet with a tail. Here it's a big tail. The tail is almost bigger than the entire cell in some of these. And we can measure this directly. Now, there are many other ways we've measured the DNA damage in RVCL, but this is one that's most visual, so I thought I should show it. So what's happening here is the DNA is breaking. It's a double-strand DNA break. Now we know that RVCL is a DNA damage syndrome. In fact, in addition to DNA breaks happening, we've seen deletions where the mutant TREX1 or the abnormal TREX1 protein that causes RVCL, which is a toxic mutant protein that's located next to the chromosomes in patients that have this disease. When you have a DNA break, here we intentionally introduced a break at a cut site in a cell. There's a big DNA deletion, almost like a bite out of the chromosome that's taken. So here we visually represented it. Here TREX1 in the wrong part of the cell, a nuclear TREX1 is attacking the chromosomes and degrading it. This discovery happened because of a partnership that we built with our collaborators in Japan at Niigata University on the west coast of Japan, the Brain Research Institute, Taisuke Kato, Osamu Onodera, and many others there who have been amazing collaborators and wonderful to work with and very open in sharing data. We independently came to the discovery that there's DNA damage in RVCL, but by merging our efforts, we were able to prove it. And now we are using this information to develop treatments. This is a fruit fly eye that they generated, our collaborators in Japan, and this lower half has the human TREX1 gene in the fruit fly. The upper half does not, and you can see the damage to the eye of the fruit fly. People in my lab like to say this fruit fly eye looks like a strawberry. I like to say it's the fruits of hard work. In any case, the key point is that the fruit flies also, when we analyze different genes that are regulating this, made it very clear that there is DNA damage at the heart of this. And in fact, we can visualize the DNA damage. We can, with fluorescent markers, show the DNA breaks, which are these bright spots here in the nucleus of the cell where the chromosomes are. And we can manipulate this. We can block it with inhibitor drugs. We can mutate the protein so that it no longer can attack the chromosomes, or we can export it from the nucleus, literally push the protein out of the nucleus, and it no longer causes so much DNA damage. We've also seen this evidence of DNA damage in patients who've done in vitro fertilization, where we've seen chromosome deletions in when they do genetic testing, and fewer crossovers. This is shuffling of the genetic deck. Importantly, it's important to know that this work happened in the course of normal clinical care. This does not harm the embryos, any of these analyses. We're talking about like 3-day-old embryos in a dish in a lab where they take 1 or 2 cells and they can analyze what's going on in the DNA, but the embryo remains intact. And so now in these particular cases, there have been deletions in the chromosomes where the embryo would not have survived due to the fact that the mutant TREX1, we think, had damaged the chromosomes. So the embryo would have been non-viable eventually. But the point is, we have lots of evidence in the patient cells, in flies, in mice, and the list goes on and on. So one of the things that we found was that women with RVCL are getting breast cancer at an early age. Not too long ago, I had a discussion with a patient who had mentioned to me that his sister died of breast cancer when she was 35. She may have had RVCL too. It's just that the breast cancer may have begun before she had ever could have been diagnosed with RVCL. And so we think that this risk might even be underestimated. Now, this does not mean, if you're a woman with RVCL, it does not mean that you're guaranteed to develop breast cancer, but it does mean that the odds are higher, and therefore the screening should probably be different. So it's important to talk to your physician, whoever that might be. And for those of you who want to talk to me, please feel free to reach out to me and I can give you more information. So now we think we understand what's driving RVCL, what's creating the risk of breast cancer. It relates to the DNA damage theory of aging, or these chromosomal injuries. And again, this happened because of our colleagues in Japan, our colleagues in Australia, our colleagues in Scotland, all working together with us, colleagues in France, from around the United States. Here we have Michaela Gack from Florida and Max Artemov at WashU, who's a great friend and collaborator who helped us with this. We published these results in the Nature family of journals, Nature Communication, where we explain how the abnormal TREX1 is causing chromosome injuries, causing increased risk of breast cancer. And we're seeing these chromosome injuries when we express the RVCL mutant form of the TREX1 gene in fruit flies, in mice, or when we look in the patient cells. 40 authors from all around the world working together, every single one of them making absolutely critical contributions. 4 different continents involved in this work. Now that we have this information, we're able to screen very efficiently for protective compounds. We've screened 7 billion compounds. We've narrowed it down to a few hundred. We have lead compounds. We're on track to cure this disease, largely because of our collaborators at the University of Michigan, who are incredible. We have chemists who are synthesizing these, but we don't have enough hands. We need more hands at the bench to be able to get this done. This requires manual labor and work, and it's not just about being smart. It's about hard work and effort. Most of all, I want to say thank you to the patients, to the families for reaching out to us. We are doing this for you. We're committed to helping you. We have a long way to go, and we want to thank you for giving us purpose in pursuing our shared goal of developing effective treatments and a cure for RVCL, RVCL-S. Thank you to all of you for everything you do. Have a good day.